Aj is the Chief Exploration Officer and Head of Neuroscience at Insitro. His team is really impactful with leading cutting-edge technologies and integrating them into their drug discovery pipelines, all in pursuit of therapies for patients. And he's going to tell a really compelling story about ALS today. Thank you, Aj.
Thanks for the invite, Jillian, and it's really a pleasure to be back in Boston, even under the weather conditions. Used to be right across the street at Novartis, so I visited the Broad many times, and it's also just really a pleasure to actually hear all the talks and be very inspired by everybody, and also humbled by the work that Casper and Kuralis is doing, and the talk that Madonna gave on kind of ALS, and I think they set it up very well. If there's a few things that you can take away from the talk I'm going to give is really the speed at which technology is moving, and also kind of the ability to harness that technology to really help us kind of diagnose patients, make drugs, run better clinical trials. So I'm going to go through. It's going to be a very technology heavy talk, but very much kind of want to show everybody the work that we've been doing at Insitro for the last about 8 years. So I'm going to skip over kind of the background on Insitro, but just quickly, end-to-end company that really focuses on causal biology, focuses on an end-to-end process to be able to develop drugs, with a heavy focus on machine learning. So about 5 years ago, we formed a partnership with Bristol Myers around ALS, and a lot of the background was already given here, from Casper. We've very much focused on sporadic ALS because it's the major population, and we've really kind of focused on very much the loss of TDP-43 and the splicing defects, which Casper already went over. I've had a pleasure of actually interacting with patient advocacy groups, and Brian Wallach here is somebody that we've interacted a lot with. He's pretty amazing. He was diagnosed with ALS in his 30s and has really gone on to kind of be a major patient advocate, and actually pave ways, with the government to actually be able to, further move therapies much faster. And so, really think we just need more people, kind of like him that are really here to kind of, make, kind of advancements as fast as we can. So at Insitro, we're a physical AI company. We've focused a lot on building automated labs. We have about 100,000 square feet of lab, and we've actually focused on automating as many processes as we have. And many people here work on stem cells, on iPS cells. They're very difficult. We've really kind of focused on automating that process and really used this for ALS. So we've kind of taken hundreds of patient lines, edited those lines to correct mutations, or taken kind of control lines and actually made edits in those control lines, and actually developed very rapid protocols to make human motor neurons. And that's what you're seeing here. Through the last 5 years, we've done this for over 200 lines, and really made kind of a process to capture the phenotypes of these neurons, really to seek can we really model ALS in a dish. And on top of that, we've developed machine learning tools to be able to look at things like cell morphology. So this is also work kind of most of know Ann Carpenter, who's actually used cell paint to look at cell morphology and use that for phenotyping. We've done that kind of at mass on these motor neurons, and actually what you're seeing here is a machine learning algorithm trained to actually be able to distinguish either control cells or mutant cells that actually are either from patients like C9orf or mutations that we've engineered in. And what you're seeing is that using kind of unsupervised learning, we can actually distinguish the difference between kind of a wild type line and a C9 line. And what you're seeing is different channels. A lot of the information actually is contained within the DAPI channel, which is just looking at the nucleus. And so it seems that the nucleus is very different between wild type cells and cells that actually have kind of mutations for ALS. And so we've very much taken kind of the information that we get from these machine learning models, something called an embedding, and ask, can we actually use that? Is it useful to do any kind of predictions? And so you can actually take that information and ask, for instance, can you predict TDP-43 localization in cells that actually are not labeled? And that's what you're actually seeing on the right here. So we actually do a pretty good job at actually predicting TDP-43. Which is we know that TDP-43 is actually lost in the nucleus of kind of patients. And so we, kind of focused on that, and we know that TDP43 is involved in, kind of defects in splicing, and so we focused on the splicing defects downstream of TDP43, similar to what, kind of Casper talked about. Except we've taken a very different approach because not only, is UNC13A changed or Stathman changed, but there are, hundreds of genes that are downstream of TDP43 that are changed. And so we've focused on doing modifier screens to see if we can actually find, cellular genes that when you knock them out can actually correct the defects in TDP43. And so to do that, we set up some platforms at Insitro. One is called optical pooled screening, which was developed by Paul Blaney. We've automated that process. And in the optical pooled screen, what we do is we actually look at motor neurons, and we look at Stathman cryptic exon along with full length using FISH probes, along with TDP43 localization. And then second, we've set up something called flex-based perturb-seq, which allows us to go beyond what you can do measuring the 3' transcripts, in perturb-seq, allows you to measure cryptic exons, and so we've actually done that, for about 40 cryptic exons. And so we've really used these models along with machine learning models to be able to, screen through the genome to ask, are there protective genes that when you knock them out will actually restore the function of TDP43 in its role in splicing. And this is just some of the automation that we've built to be able to do this at scale. So, in addition to all of the automation, we've developed machine learning tools, and we really like to kind of take the human, out of the analysis of cells. So we do something called self-supervised learning, where we actually ask, kind of the algorithms to actually determine, what's going on in an experiment. That's actually hard for people to understand, and so we've actually done something where we actually kind of disentangle the information and actually make it interpretable to humans. So what you're seeing on the right is actually one of these interpretations where you can look at, kind of Stathman cryptic exon in green going up, or the cell size changing, or TDP43 mislocalizing. And these are things that are just completely learned from, from computer vision, from machine learning, not from a human, completely unsupervised. And it actually starts to come up with, kind of things that a cell is doing, which is actually very useful for us as scientists. You can actually do very interesting things with this data. So you can ask, which cells look like, each other. It's kind of like saying, which people are in families. And you can do things where if you have, kind of control genes, you can actually say, kind of, what are all the genes that actually change the TDP43 phenotype from, being missplicing to actually splicing, and that's what you're seeing, kind of here on the right. And you can also ask, what are genes that kind of, will further make the phenotype worse. And so this is really, kind of looking at, can we really model disease in addition? Can we actually find genes that actually modulate TDP43 in this very critical splicing, kind of role? And so this is just, kind of iterative cycles of the screening process of the optical pooled screening and perturb-seq to look at, what are human genes that when you knock them out can kind of protect for TDP43 loss of function. And what you're seeing here is just some plots, kind of looking at rescue of TDP43 along with, because this is an image-based assay, you can look at TDP43 localization. Along with, on the right is kind of something where you look at the cryptic exons versus, kind of full length, and we've kind of made scores because it's about 40 genes that actually make this up. And the nice thing is there are multiple genes between these two different assays that actually, kind of rescue the TDP43 phenotype, and it, corroborate each other. So one of the things that we've been really implementing, kind of recently is, kind of, and I think many people are doing this, is kind of using agents to kind of help us sift through data, right? So, there's a lot of data out there in terms of all, papers, there's, knowledge graphs, there's our kind of internal data, there's clinical data, and it's very hard for any one of us to kind of, actually understand that data and cut across that data and actually make any sense of that data. And so this is a project, where we've actually generated agents. Kind of there's an orchestrator agent, kind of the project team lead that actually organizes all of the other agents. And within Insitro, we like to have data which is kind of called ClinML. That's kind of human level data, so human genetics or other human level data. The CellML data is the kind of data that I showed you, which is kind of in vitro disease modeling. along with kind of literature and knowledge, kind of graphs or things that you can actually bring in. And so we've been integrating all of this to really ask, kind of, what is a good target for ALS? And you can do things where you turn off our data or add additional data in there. And it's been very interesting. We had initially with BMS, we actually went through all of this, kind of with humans. It took us a couple months to kind of, go through all of the literature and come up with, kind of a hypothesis around, hey, this target is something that is, very useful for ALS. This will do that in about 5 hours. Kind of, and it actually comes up with very similar results, and actually will even suggest the next experiments that one should do. And we go back and we actually look, and it's actually the experiments that we did. So it's actually really, kind of quite amazing how kind of technology is now speeding this up. I don't really think of this as something that replaces humans. This is something that just helps us. It's kind of augmenting us. It's kind of like how everybody is using LLMs now. Everybody uses ChatGPT to kind of augment. And so I think this is really an opportunity to kind of lean into that to kind of speed up, a lot of the drug discovery process that we're doing. So just a bit on kind of the targets here. So, one of the targets, and unfortunately, this is a collaboration with BMS, and so I'm trying to get these targets published, hopefully within the next year, but they wanted a little bit of a head start on actually working on these. So, siRNAs to these targets, kind of are well tolerated in cells, and kind of just show that on specific cryptic exons, these actually revert the cryptic exons kind of down to kind of wild type levels. When we look at more cryptic exons, you can see that kind of Stathmin and UNC13A are there, but there's many other kind of cryptic exons and full length genes that are important for TDP-43 function. And so really hoping to learn from what kind of Curalis and Casper is doing, and I'm really hoping that those things kind of go forward, but hoping that we can learn from that and kind of further advance some of these potential targets. A little more on kind of the knockdown of this first target, rescues Stathmin protein, not just RNA, and so that's something that is really kind of nice nice to see. So really to kind of wrap things up, we've really focused on using data science and machine learning to really help enhance drug discovery. We have a whole additional work to kind of credential, and actually similar to work that others have shown on kind of neurite outgrowth. Actually, these targets kind of restore neurite outgrowth in human motor neurons. That's what you're seeing in the right, and so. Hopefully at a future time I can come back and talk about the drug discovery efforts that we've done. So we've actually started to move forward, both on oligos and small molecules on these targets, and we'll be moving these forward with BMS. So, but yeah, really just wanted to highlight kind of how fast technology is moving, and how we can use technology really for rare disease. Insitro, this is last year, so yeah, a lot of dedicated people, kind of working here to kind of, bring things forward.
I don't want to be the only person asking questions. Obviously, I love this type of work. My question is, TDP-43 is a stress protein. And so my holy grail for ALS has always been, well, what if you just can prevent TDP-43 pathology altogether? Yeah. Do you have any feeling that that is a possibility? And because you had a lot of stressors, by doing these type of screens, we know there's ER stress that can trigger TDP-43 mislocalization, hyperexcitability can trigger it. There's autophagy stress that is associated with TDP-43. Did you get a new biological understanding? And tied to that question, which I always find fascinating if I see large screens like this with sporadic lines, do you think all of ALS is genetic?
Yeah, all really kind of interesting questions. I'll kind of start with the kind of stress biology. I mean, there are interesting things that came from our screen, kind of in terms of those pathways that kind of come up as things that are kind of reverting the phenotype or enhancing the phenotype. So I definitely think that's a role. I think it's still, kind of hard to know where the stress is coming from. Like nobody knows where, kind of the drivers of TDP-43 dysfunction are really coming from in terms of, a patient. So it's kind of hard to know, kind of where to intervene there. And I think many people have different ideas in terms of how that happens. Around targeting TDP-43 itself, I mean, there's definitely companies like DuPont, which are actually have kind of cell mods that are trying to disrupt the TDP-43 aggregates. I think those aggregates are really hard to disrupt, and kind of to have a brain penetrant molecule is going to be really challenging. But the data they have is pretty good. So I'm really excited to see those kind of moving forward in terms of all of these things we do are kind of experiments, and we need to get these into humans and try them, and that's really the only thing that matters. And so I think, kind of we'll see as their kind of cell mods move together, whether disruption of TDP-43 aggregates will have an effect.
So just so you know, we've tried to do something like you did, but in a more primitive fashion. And we ended up getting a lot of targets that were already identified by other academics. And then we thought about doing the DuPont approach, but then we worked with Manuel Navia. He's one of the structural biology experts in this environment. And he said, well, you're probably going to end up with a lot of soapy compounds that you can never develop into real drugs. And so that we didn't go that direction.
I'm surprised that the DuPont molecules are actually kind of holding up.
They're targeting something else, I think. But yeah, I'm rooting for everyone that is going to do something about this disease. So hope
yeah,
we're successful.
Me too. That's why I'm just excited as the whole field moves things forward. And we're just going to learn from each other.
Exactly. Yeah. Thank you.